Next, you also have something that's negatively charged called a cyclophosphorace. Okay, more a bit like I would say like a pseudo sugar molecule here, but the difference is you have your sulfonal group, right? Negative charge, your SO4 negative. You see the reason why I put carbohydrates because right, if you substitute this with OH, it is your actually like a sugar. Right? So this is really some kind of a post-modification. You think you can convert your OH to a sulfonic group? Okay, here it is. Okay, next we have ionic liquid. As we talked about it just now, that these are very special compounds that it has both the charged species, charged group, and also the hydrophobic tail. And therefore, as a whole, you realize that this is unlike ionic salt like sodium chloride, few hundred degrees Celsius for melting point. This liquid melts below 100 degrees Celsius, or sometimes even lower than RTP. The reason is that the primary interaction comes from the dipole-dipole. In fact, it's induced dipole, IDPD, your IDID, your London dispersion forces, and of course, it still possesses some of your ionic attraction. But as a whole, which is stronger, it is because of the tail, right? And this tail are hydrophobic. So think about it: you have a tail, you have a charged part, right? You have duplicate of this. If you want to harness your Coulombic attraction, right, you need all the charge to be together. But because of space dispersing them, so it's not really like a full classical ionic structure. But it's like you have a mixture of ionic interaction and your London dispersion forces. So therefore, it is ionic, but yes, a liquid. But the good thing is, right, that means it shows properties of more simple covalent structure and also ionic structure. But what we want them for is really like a solvent. It is stable and it shows solubility in both the polar and non-polar solvent. Here, another classic chiral selector, we use a monolith. You can see we use a SEM, scanning tunneling microscope, electron microscope to see. So these are polymer-based monolith, like a tubing. We can explain it like using a column made of like polymer with large blocks. So if you want to use an analogy, is think of it like a carbon nanotube. You have a flat layer of your graphene. Graphene came from just one layer of graphite, and then you take one piece of graphene like this, right? Okay, and then you roll it up this way, like a nanotube. So in a way, monolith is like the same fashion. Now on the left here, how do you do some modification, right? You want to make your monolith surface functional, so you have to do a modification. In this case, you have a complex molecule telluride, and you have a primary amine group doing a nucleophilic attack, a ring opening of the epoxide to form a hydroxy amino group here. In this case, you form a larger chain. Okay, so this is a bit of organic chem, right? And you see that, right, there are three chiral centers in this telluride. Here, put in the stars. Hope you still remember stereochemistry, which is your R, S, and R. Okay. More examples here. This is your particle fixed monolith here. So we are trying to separate this compound, which is a polyamide made of two amino acids, your glycine and methionine here. So the chiral centers here, you have both your R and S. So how do you separate them? We use a capillary electrokinetic chromatography on a particle coated or loaded tecoplanine alicone monolith. So you see that here the peaks looks a bit different. Okay, a bit different here. One is sharper, one is broader. Okay, but the point is that we would like to have a clear separation of both. Next, this is another new innovations. Not too new now, but still something you want to think about for your research next time. CE is also portable. You can bring a small equipment like a suitcase and go to on-site analysis. chiral separation using CE is possible. We call it the miniaturized technique. Have a substrate, again with epoxide, and then your enzyme. So this is a ring opening to form a diol in this case. So what you do is put this substrate inside this channel, your catalyst in the other channel, this is a natural enzyme catalyst. Then you run it in this monitoring system, go through a pathway, undergoing catalytic reactions. And here you have your SO sample outlet, your buffer inlet, your alternative sample inlet here, and you run and then you see that then you have reactions and separation at the same time. The reason is that here you have a chiral center in this carbon pointing to, so you give rise to two enantiomer. Right here, so we continue with this chiral separation using a miniaturized technique. This is a shortened single wall carbon nanotubes here for the separations of the bovine serum albinase conjugate. So you can see this is more like a synthesis scheme. So start with first of all this nanotubing, and then you have addition of acid refluxing to purify the surface internally, and also adding the 2 carboxylic acid group here. After that, we do something called the cutting to slice so that this tube won't be so long here, to be like the shorter chunks here. So you can imagine this is similar to like cyclodextrin, right? Shorter, smaller molecule. And then after that, we added this EDAC, the 1-ethyl-3,3-dimethyl-aminopropyl carbodiimide here, right? To change the functional group modification. And after additions here, we have a chiral selection. Okay. In this case, we continue with showing how we combine this ITP CZE here. Now the top part indicates a sample pre-concentration and how we remove the impurities, right? And the bottom is the CZE with a higher resolution and separations. Now notice that for A and C right here, this is the chiral separations of the DBP and LBP, right? And we compare this way here. The B and D are the chiral separations of the phenylamine, the PHM1 and PHM2, and the bottom side, right, is similar here, right? So when we combine this ITP and CZE, the switching time from the ITP and CZE needs to be optimized there, right? And you see that this capillary zone electrophoresis does show some separations of the chiral isomer, right? Because there's a lag time between the two, right? And here we have this blown up the zoom window to show where the chiral isomers are detected. Next, so during the chiral separation by C, right, there will be an interconversion reaction. We call it the enantiomerization reaction that takes place. And here you see that, right, it's interesting here. This is not the baseline. Baseline is here, right, across the baseline, but it's a plateau. And this plateau, what it tells you is, right, it consists of a mixture of both the separated enantiomer, right, over here, because it's an equilibrium. So it is really the equilibrium between the forward and the reverse reactions where you are trying to enantiomerize. And there's an interconversion. So we can have a parameter to use to characterize the peak pattern, right? HM by this formula is based on the height of the plateau here, the height of the plateau, right? And also the peak heights of the two enantiomer peaks here, okay? The V1 and the V2 there. So this top trace, right, shows the experimental result, and below here it shows the simulated result. And if you can do some simulations and then calculations, it is really possible to determine the rate constant for this interconversion between the two enantiomer. Okay, very interesting. Next part, we also introduced to you what is some miscellaneous technique here. You know, normally for C, right, it is done by applying voltage on both ends of the capillary. Now here you see some innovations. You can also apply voltage around the surface of the whole capillary to enhance the separation. And this method is based on this radioelectric field. Okay. The advantages of this is although it's quite unique and less mainstream, is a new separation mechanism can be introduced. So basically in research, we have a hypothesis, we try and then see, oh, it can separate with better efficiency or shorter time or even slower time, but because the construct of the infrastructure is different, we can still publish something and let people imagine what can be done in the future. Now, the disadvantage is that, right, this instrument is more complex because, compared to the two ends we apply voltage, now you have it around in the middle. So you need to have a bit more complex engineering. This is another example of showing an electrophoregram of the two different enantiomers. See the R and the S there? Okay. Now again, how you see cathode separation is notice the alpha CD cyclodextrin. Again, try to look at this the subtlety in the informations, right? You can see what is the voltage, what's the conditioning temperature, what's the buffer system, the internal diameter of your capillary, but small things like having CD tells you about your substrate being chiral, so you can tell me that, oh, is it 1, 2 are enantiomer, or 1, 2, 3, 4 they're all different compounds of the same nature. Okay. Here we're trying to show that, right, although a CE doesn't need a high amount of your substrate injection here, but if you can dilute, right, you see dilute 10-fold, 20-fold, 40-fold here, it is trying to test the detection limit. What you see for A, right, without dilution, the noise, signal to noise ratio is very good, more than 3, but as you slowly dilute further, you see there was a peak here, then now the peak is like a noise, so we can't even see it. So this is not what we want, but of course we can still do baseline later, but it might be less reproducible. In this case, similarly, right, we do environmental analysis using current separation. You see that, right, look at the label A, B, C, D, okay, this order. Now for A is, right, you have this the analysis of this duloxetine and iconoazole racemic from wastewater. A is just in aqueous form, okay, there's no reaction. Then for B, it is a starting time where we put in the calcium medium for a biological reaction there, right? So at start is again pretty much similar, but after some time, 72-hour incubation, right? Abiotic condition, you see these signals are gone. Iconazole disappeared. And right, if you do the same reaction, but in the presence of this Daphnia magna is like those little worm there in the water, right? Then it changes also the amount of the duloxetine. So it's very interesting. Again, I want to highlight here this S stands for sulfate beta cyclodextrin. More data. Okay, for drug addicts, right, we need to test whether they are taking drugs. So here we have some forensic analysis of ketamine and not ketamine. Okay, from hair sample, you pluck the hair, you look at the follicles, and then you snip it, and then you grind, and then you prepare the solution. Then you can see and ascertain whether they are still taking drugs or are they lying to you. Okay, and of course for each of these ketamine and non-ketamine, you have also the enantiomeric form. Important to look at the amount of these drugs in the sample. We call S stands for sulfated. Next, what are the factors affecting the peak shape? Very important, because if you don't have a very good shape, right, then your peak area won't be consistent. There'll be error involved in elucidation. Now, one thing is, right, it's about power dissipations. We know that this capillary tip is round and spherical, and it's round, so it contains actually an electrophoretic medium, right? What it means is, right, you can think of it like it's like a copper wire, because copper wire inside is copper 2+ ions, right, all closely packed with electrons moving, right? But in this case, it is similar. You have a capillary that's hollow, but because what's flowing inside are your electrolytes, so in a way it's similar to a copper wire. So you have V equals IR, we can take it like a reference. So your potential difference is your product of the current and the resistance. So to achieve high efficiency in CE, right, we need to ensure that there's no heat being trapped inside there, become a hot spot. We need to ensure the sufficient heat dissipation. Okay, and how do you do it? We refer to this equation there, right? So just know what the the acronym stands for the P is power, right, over the length, and C is the buffer concentrations, V is the applied voltage, right? So all these variables will have an influence on your resolutions of your peak, your CE. Okay, in this case, right, I want to just say that when you have a possibility of adsorption because of larger protein, it's lower mobility, you just stick to the surface there, right? What it does is you have a peak distortion, and when you have that, it's not very reproducible, which means you can't use it as a reference to see whether you have substrate A, B, and C there. You can't even use it. So, and how it arises is, right, it stems from the larger plate height. If you have a large plate height, right, we know that it is synonymous as your lower separation efficiency. We want to have lower plate height so that we have more number of plates for better separation. Okay, so this is the equation to determine whether your separation is efficient. Next, so what are the common peaks that we do not desire, right? So we have something called the fronting peak. Fronting means do you have a certain ions with a slightly higher mobility than those that move in the band, right? So that means when they come up a bit faster, the detector will see them coming up first. That's why you see a bit of peak, it is too kanjong, too fast to come up first, and that's why here it's very sharp, right? We don't want that. And on the other hand, you also have something called tailing, which means that there are a few of these ions is a bit of maybe bulkier, floppier, or it's a bit heavyweight. They come up slower. So even though your band of sample comes up first, but there's a bit of tailing because they're still being scanned by the window. Okay, so tailing, or both are tailing and fronting are not desired. Now when you don't have such an effect, you have a very symmetrical peak, which is so desired. If you ask me which is more common, it is more tailing effect. Okay. So in summary here, we have gone through different modes of CE, and also we talk about how the additive in terms of the CD, in terms of the crown eater, how they complex and using the different rate of mobility, right, and affects the separation of an enantiomer. We talk about how we can estimate power dissipation, what's important, and the effect of adsorption on peak height, peak the shape and efficiency, and of course we're trying to match the mobility to get a good peak shape. So with that, I will end off this slide, and I'll see you in the next lecture. Cheers.